Okay, welcome back. And so what we're going to progress on to now is looking at, in more detail, aspects of the adaptive immune response. So today we're going to be talking about T-cell immunity, and Kim's going to help me with that. And so where we are now is that we've learned all about the nuts and bolts. We've learned about antigen receptors. We've learned about T-cell development, MHC, processing, where it happens. And so now it's bringing them all together in the next series of lectures of how we generate these responses. And so it's broken up into T-cell immune responses, and then after that we'll talk about B-cell immune responses. So to start things off, Kim, T-cells, are they all the same?
T-cells are different. We've learnt they've got different receptors, but we also can group them into different types. And as technology has gotten better, we've learnt about more different types. But if we break them down into the basic types, we have our CD4 T cells and our CD8 T cells and our regulatory T cells. So our regulatory T cells we've learnt about when we learnt about central tolerance, the policeman of the immune system. CD8 T cells are also called killer T cells, and so they're good at directly killing cells when necessary. And then we have our CD4 or helper T cells. And we can then break these down into further categories as well, of which one of those categories is actually the regulatory T cells. And they all have slightly different functions within the immune response.
Now you just mentioned T helper, CD4, CD8. How important is it to actually know what each of those means?
You should understand that a helper T cell, if I talk about helper T cells, you should know they have a CD4 molecule as part of their T cell receptor complex. I think it's really important to remember those distinctions, because people will use helper T cells or CD4 T cells expecting you to understand that they're the same thing.
Yeah. We don't do it to confuse anybody, but often we'll read it that way, and so it's really important to understand what each one of those means. Why are there different types of T cells?
We have different types of T cells because we're infected with different types of pathogens. So a viral infection will infect a cell. The pathogen is inside a cell. Lots of bacteria will be outside of the cell. So we need different ways of dealing with those things, and the different T cells are able to do that.
And so does the immune system target a particular type of pathogen when it makes a response? For example, will a particular immune response only make CD8s?
No. So you'll always have, we talk about viral infections and that you get a CD8 response, and we sometimes forget that you also have a B-cell response and you have a CD4 T-cell response. It's a really integrated system. So the CD8 T-cells we also call killer T-cells, and they can directly kill an virally infected cell, for example. But it can't do that without the help of a CD4 or helper T-cell, which will provide it signals to tell it is allowed to kill an infected cell.
In essence, an immune response is a fairly generic process, and it's only the little details that change depending on what you need to target. Is that right?
That's right. So each time it's a virus, you're going to have a big involvement of CD8 T cells.
Yeah. So if we don't understand that, then we're halfway to understanding the immune system, right, yes, or the immune response. We've sort of touched on it, but do all T cells have the same effector mechanisms?
So effector mechanisms are what a cell does to get rid of a pathogen. And no, our different T cells will have different effector mechanisms. So CD4 or helper T cells will help other cells of the immune system to respond strongly, respond appropriately. So, for example, they can produce cytokines that will make a macrophage better at phagocytosing and killing a pathogen. CD8 T cells, or cytotoxic or killer T cells, will kill cells that are infected. And regulatory, or CD4 positive, CD25 positive T regulatory cells will often increase at the end of an immune response to help dampen it all down, so that we don't have this chronic inflammation, which would obviously cause us ongoing pain. But once we get rid of a pathogen, we need to stop the immune response. And
I think earlier on I mentioned that T cells sort of develop in the thymus, okay, and so most of them die, and then whatever's left gets released. So are they ready to go at that point, or?
When a T cell leaves the thymus, it's known as a naive
What does that mean?
T cell. So it hasn't encountered its cognate antigen yet. So the antigen that it will recognise, it hasn't come into contact with it. But it is mature, so it's a naive, mature T cell. Once it has come into contact with its antigen, which has to be in the context of an MHC and co-stimulatory molecules, which students will have learnt about by now, those interactions will turn it from a naive T cell into an effector T cell, and that's when it can start to have its effect on other cells.
Where does it actually see the antigen?
So we have so many T cells with different receptors. The chance of just randomly coming into contact is quite low. So they go into lymph nodes, and that's where the interaction with an antigen is going to occur.
So the antigen gets transported through the lymphatics to the lymph node and interacts with the T cells there.
Yes.
Okay, so the T cells are really important for generating the immune response, and then they'll go out and do stuff, I suppose. They'll go out and kill cells that are infected with viruses or interact with macrophages or whatever. So every time we make an immune response, do we just keep those cells and make the next one?
So at the end of an immune response, we really need the number of T cells and B cells to shrink back down. So we don't have all these T cells that are exactly the same, because you think the body always tries to maintain homeostasis. So there could only be a certain number of T cells. And at the end of an immune response, you don't want to keep all that respond to this virus and all of a sudden you can't respond to the next virus. So the immune response will shrink once that pathogen is no longer there to stimulate making new cells. But we don't get rid of them all. And this is a really important part of the adaptive immune system. We keep some as memory cells. And that means if you're exposed to the same pathogen again, those cells are already there. They don't need to be activated in the same way that a naive T cell needs to be activated. And therefore it's ready to go, and your immune response is much quicker and it's much more powerful. So you can get infected but not get sick from pathogens often when you're exposed to them a second time. And that's the result of these memory cells.
So if we get infected with something, how many of those cells would actually be useful, do you think?
You'll have pathogens which have multiple antigens on them or inside of them, and multiple T cells that will recognise them. And the best ones will be chosen and will expand preferentially. So that's what will happen in the lymph nodes. Once they've recognised an antigen, we'll make more of those T cells so that we can have enough to mount an effective
So we
immune
make
response.
a specialised army.
We make an army,
Okay.
and at the end we disband the army and keep
some.
a few sentinels around.
Okay.
Just
All right.
in case.
Cool. I think that's pretty much it. Thank you, Kim.
Thank you.